Hello. In our last screencast, we looked at just kind of some general principles of energy in living things and how the laws of thermodynamics and chemistry and physics all kind of play a role in what happens in living things. So, with this next screencast, we are going to get into the nitty-gritty of what actually happens when a molecule of glucose is broken down to produce ATP. So, let's dive in. Um, Before we get into that, there is a handout that might be of use to you. This handout is it's called Overview of Cellular Respiration, and either you picked it up in class or you can also access this on Schoology. It is part of the study guide and embedded within those outlines of the study guide. So you could print it there or pick it up from class. But we're definitely going to be using that today. So, quick overview of this process called cellular respiration. So the general idea of cellular respiration is we are in accordance with the first law of thermodynamics, converting one form of energy into another. And in this case, both forms of energy are chemical energy. So we are converting the chemical energy of food into the chemical energy of ATP. And you may think, well, why? What's the point? If they're both chemical energy, why do we need to do that? Well, remember, food is kind of like that $1 trillion bill, right? It's got tons of energy, but it's not very useful for day-to-day transactions. If you want to go to a movie, you're probably not going to take a $1 trillion bill. If you want to order something from a vending machine, probably not going to slip a $1 trillion bill in there. ATP, however, is like a 20. And so a $20 bill is very useful. And so in this process of cellular respiration, what we're doing is we're taking that $1 trillion bill and we're breaking it into lots and lots of 20s that can be used for day-to-day cellular processes. And again, there's a long, complicated series of steps that we're going to do to convert that $1 trillion bill into 20s. So we're we're producing energy in the cell. Which organelle would you expect this to occur in and why? And then which monosaccharide would you expect to act as fuel? So hopefully you know the answers to these two questions because of our past units. So in Unit 2 on cells, we learned that the mitochondria, in fact, is the organelle that produces energy. And lo and behold, the mitochondria will feature prominently in today's, the next screencast or two. And then the monosaccharide is glucose, which again, hopefully you memorized in Unit 1 that glucose produced or is used to produce energy for the cell. And back in Unit 1, you just kind of memorize that fact. Glucose makes energy. Now we're going to understand how that actually happens. So our overall reaction is glucose, C6H12O6, plus oxygen, 6O2, yields 6 CO2 plus 6 water, and then that gives us that ATP. This is an equation that I think is worth memorizing. C6H12O6 plus 6O2 yields 6CO2 plus 6H2O, because it comes up I don't know, it's just useful to memorize it. So you might want to log that one in there at some point. So the process is quite complicated, and so we're going to break it down into a series of steps. Glycolysis, pyruvate oxidation, which is kind of this unlabeled intermediate step here, the Krebs cycle, and the electron transport chain. Now on this sheet, this handout, you'll see the table below, and this table below kind of summarizes what's happening in each of these major steps. And I'm not going to go through that right now, but this is kind of a good reference tool for you to come back to. So let's begin with that first process, glycolysis. Glycolysis literally means sugar split. Glyco, sugar, lysis, split. And what glycolysis does is it partially breaks down glucose to harness a small amount of energy. Okay, partially breaks it down to release a small amount of energy. It occurs in the cytoplasm of both eukaryotic and prokaryotic cells, and the net reaction is right here. Glucose is converted to 2 molecules of something called pyruvate. And in the process, we get some ATP out of the deal. Remember, that's our $20 bill. That's what we're trying to make is energy. And we get some of this thing called NADH. Now, hopefully you remember from our last screencast that NADH is one of those electron carriers. It's kind of that intermediate sort of holder of electrons. I kind of had that little symbol on the hill where it kind of, instead of just pushing the electron all the way down the hill, NADH can kind of grab it midway through the hill and kind of pass it on to something else. So these electron carriers are going to feature prominently. So for now, it seems a little random, like what's this NADH, but just trust me, it'll come back and make sense later. So basically, we get a little NADH out of this and a little ATP. And you might think, well, Mr. Sanger, a little is kind of a vague term. Can we quantify that? Yes. And so again, on this handout, you'll see this table, and you can see that in this overview diagram, we see that same thing. Glucose going to pyruvate, 2 molecules of pyruvate specifically. We get some ATP out of the deal. We get some NADH out of the deal. And then if we look at this table, we can see the specifics. So we specifically get 2 molecules of NADH, and we specifically get 2 molecules of ATP. Now, this third column, we're going to ignore this for a little while because ultimately our NADH is going to be used to make more ATP, but that doesn't happen until a later step. So for now, we're just going to ignore this entirely and focus on this NADH, 2 molecules, and this ATP, 2. Okay, so that's the overview, but what does that actually mean? Glucose to pyruvate, blah, blah, blah. What's actually going on? Well, you're going to regret asking because it's complicated. So here is the diagram, the figure that shows all the major steps of this process of glycolysis. It is a catabolic pathway because it is breaking down things. And you look at this, and it is very normal to be intimidated and freaked out a little bit. That's okay. We're going to break it down and figure it out. A couple points before we get into it. First of all, remember the analogy we used last unit. The cytoplasm is like a ball pit. So molecules, including water, are all just kind of bouncing around inside the cytoplasm. And so when you see random things like, hey, this ATP comes out of nowhere and interacts, where'd that ATP come from? Well, that ATP is just floating around in the ball pit. The cytoplasm has tons of stuff. So when things seem to randomly appear, it's because they were in the ball pit the entire time. Second, every reaction taking place has either a negative delta G, meaning it can occur spontaneously, or is going to be coupled to another reaction with a negative delta G. In other words, we still need to follow those rules of thermodynamics. We have to make sure that the reactions can occur. So it's either got a negative delta G to begin with, or it's coupled to a reaction with a negative delta G. Third point, each reaction is catalyzed by an enzyme. So enzymes are going to lower the activation energy so that a reaction will occur. And you might think, well, where are those enzymes? Well, they're in the ball pit. They're all just bouncing around, floating in there and doing their thing. If you want the names of the enzymes, they are listed in abbreviated form right here, Gly1, Gly2, Gly3, and then right here you can see the long name. Gly1 is hexokinase, Gly2 is phosphoglucoisomerase, Gly3 is phosphofructokinase, and you don't need to memorize any of this, so don't freak out or panic. You don't need to know them, but know that there are enzymes and they have names, and note that they all kind of end in "ase," as most enzymes do. Next, the process is inefficient. Only about 3.5% of the energy originally in glucose is actually harvested as ATP. So this is not the most efficient process in the entire world. Not to worry, we will do better before the end of this unit. And then don't memorize this. You do not have to memorize it. The AP test will not require you to have this memorized, but you do need to understand the concepts. Or if you're shown like a segment of this, you need to be able to understand what's happening in that segment, but you will never be asked to just recreate this diagram from scratch or to memorize the order of these molecules or anything like that. So don't panic about that. Okay. So if we break down glycolysis, what is actually happening? I would encourage you, I have these red boxes and lines and arrows. I would encourage you to use this handout again to take notes on this and kind of label as it's labeled on this diagram. So the first process here is, the first thing that happens in glycolysis are these first couple reactions here, these first 3 reactions. And what's happening here is priming is occurring. And what that means is we're kind of getting things ready to go. The goal, remember, is to take this glucose and convert it into 2 molecules of pyruvate down here. And so these first few reactions are there to kind of get it going. You'll notice that we actually are investing ATP. We're using an ATP to add a phosphate group here, and then we're using another ATP to add a phosphate group here. So before we even get going, we're actually investing 2 molecules of ATP. The whole point is to get ATP out of this process, but we're putting 2 in to begin with. So already we're behind the curve a little bit on this. And so these processes are called substrate level phosphorylation, which means that we're just sort of directly using ATP to add this phosphate group on there. Okay, next is cleavage. So this reaction here, what's happening is this 6-carbon molecule is broken into 2 3-carbon molecules. And then this one right here is basically converted to this thing. So by the time we're finished with this step, our original 6-carbon glucose has been broken down into 2 molecules of glyceraldehyde 3-phosphate. So we have 2 3-carbon units instead of 1 6-carbon. What that means is that everything that happens from here on down is going to happen twice. And again, I would label your diagram with these little red markings. So everything here is going to happen twice because it's going to happen once for each of these 2 3-carbon molecules. Okay, next is the energy recovery phase. So we've already invested 2 ATP. We're in the hole for ATP. And so next we're going to get some actual energy out of this. We're going to get some ATP out of the system. And for this process, we need to use what is called substrate level phosphorylation. This is a process. Oh, I think I misused that term earlier. I'm sorry. Previously, that was not substrate level phosphorylation. Substrate level phosphorylation is this. It's where an enzyme directly phosphorylates or adds a phosphate group to ADP to form ATP. And so you can see this here. This enzyme has this little phosphate group, and it's just going to transfer it to ADP to make ATP. Note that delta G is positive 7.3 kilocalories per mole. And so we need an exergonic reaction to couple with this one to make this happen, because to make ATP requires 7.3 kilocalories per mole. So this needs to be coupled with a highly exergonic reaction. And so we see substrate level phosphorylation happening in these later steps, these energy recovery steps. And so as we go through, oh, here's one right here. There's an ATP, but remember, everything from here down happens twice. So there's actually 2 ATP being formed there. And then here's another one, 2 ATP being formed there. And so I would put a little 2 in front of that and a little 2 in front of that. So in terms of our ATP, we invested 1 ATP, 2 ATP. So we're negative 2 ATP at this point. But then we gain 2 ATP and 2 more ATP. So we gain 4. So we lose 2, we gain 4, and so the net gain is 2 ATP. Along the way, we also make a little bit of this electron carrier NADH. And so again, I would put a 2 in front of that to indicate, okay, we got 2 NADH. So, so there's a little video that kind of shows this if you'd like to see it. But let's just go back to original. We said that we would take glucose, break it down to 2 molecules of pyruvate. Check, we showed that. We said we'd get ATP and NADH out of the deal. Check, we did that. And we said specifically we'd get 2 NADH and 2 ATP out of the deal, and check, we did that. So we have now sort of reconciled this overview diagram with this highly detailed specific diagram to kind of account for what's happening, okay? So that's basically the process of glycolysis. And again, it's pretty amazing to think about all these molecules just bouncing around in the ball pit, and this substrate bounces into the appropriate enzyme, a reaction is carried out, and boom, turns it into this molecule. And then this molecule bounces into the right enzyme, boom. The other thing to keep in mind as you sort of wrap your mind around that, remember, it's not like there's like one glucose molecule in the entire cytoplasm. There's going to be millions of glucose molecules and millions of these and millions of these, and they're all kind of bouncing into each other all the time.